Hello everyone, I am Dr. Tarun Kumar Patel, Assistant Professor Biotechnology from Sant Guru Ghasidas Government PG College, Kurud, District Dhamtari, Chhattisgarh. Today we are going to discuss about bioinformatics basics in this computers in biology and medicine. So let's see first the introduction. The term bioinformatics is basically a combination of biology and informatics. Bioinformatics is the science concerned with development and application of computer hardware and software to the acquisition, storage, analysis, and visualization of biological information. Let's see some brief early studies in this area. Bioinformatics developed in the wake of generation of amino acid sequences of proteins and nucleic acid sequence of DNA. In 1962, Kandel and Pauling proposed that amino acid sequences of proteins may be used to study evolutionary relationship among organisms. This proposal was based on the observation that amino acid sequences of homologous proteins, the proteins having similar functions, were similar. This initiated a new field of study called molecular evolution. Subsequent researchers have permitted inference about evolutionary relationship from comparative analysis of amino acid sequences of functionally related protein. These analyses become possible due to the development of quantitative method for sequence comparison. The management and analysis of rapidly accumulating sequence data required new computer software and statistical methods. This attracted scientists from computer science and mathematics to the fast emerging field of bioinformatics. As a result, a variety of methods and tools have been developed that greatly facilitate management, utilization, and dissemination of biological information. This is one of the major advantages of bioinformatics. Then let's see the components of bioinformatics. The first component is the database. Development of database for an efficient storage, access and management of large body of various biological information is necessary. Then the second component is the algorithms. Algorithm is a finite sequence of well-defined computer implementable instruction typically to solve a class of problems or to perform a computation. Algorithms are always unambiguous and are used as specifications for performing calculations, data processing, automated reasoning, and other tasks. The development of new algorithms and statistics for assessing the relationship among large sets of biological data, for example, DNA sequences data. Then third component is the analysis and interpretation. The application of these tools, the databases and algorithms for the analysis and interpretation of the various biological databases is included in this part. Then let's see a brief introduction about the biological database. Biological database are basically the collection of life science information collected from various scientific experiments, published literature, high throughput experiment technologies, and computational analysis. The database may contain information from the various research area including genomics, proteomics, metabolomics, microarray gene expression, and phylogenetics. The information contained in biological databases include gene function, structure localization, both cellular or chromosomal, clinical effect of mutation, as well as similarities of biological sequences and structure. Nowadays, these databases have become a crucial part of modern biology. Then let's see some examples of the various databases. The nucleotide databases, the main nucleotide databases are GeneBank, EMBL, and DDBJ. Similarly, the protein databases are the SwissProt, PIR, or ProCyte. The molecular structure databases are PDB and SCOP. And other databases like KEGG, KEGG. These are some of the example of the databases. Then let's see the role of computers in biology. Millions of base pairs of DNA sequences are now known and must be analyzed. Hundreds of restriction enzymes and cloning vectors must be kept track for work in genetic engineering. Computational systems have become necessary tool for acquisition, retrieval, manipulation, and analysis of this information. In the early days of computing, it was believed that computers were excellent number manipulators, but poor at working with other forms of information. The truth which has emerged after the introduction of artificial intelligence is that computers are excellent general purpose symbol manipulators. Numbers are also a type of symbol, but so too are nucleic acid sequences, restriction maps, and even more complex forms of knowledge like strategies for carrying out recombinant DNA experiments. Then let's see the application of computers in data collection, assembly, storage, and retrieval. The current application can be broken up into the broad classes of computational tools like the data collection, assembly, storage, and retrieval, nucleic acid and protein sequence analysis, restriction map generation, simulation of molecular processes, and experimental planning and debugging. An important requirement for all scientific research is the ability to collect and store the primary data, often consist of nucleic acid and protein sequences. Computer programs are now used to facilitate all aspects of sequence data collection, verification, and correction. These programs include a variety of editing systems that take advantage of several smart display terminals. In addition, specialized tools for sequence entry have been developed, combining the overlap into a single consensus sequence. For DNA sequencing, researchers must combine the result of many sequencing gel to determine a single long sequence. This involves finding overlaps, choosing the best of these, allowing scientists to verify or correct their primary data, and combining the overlap into a single consensus sequence for the molecule being studied. Several computer programs have been developed to automate this extremely time consuming process. The other application is the information retrieval. As the database grows, the information retrieval problems become greater. Computer programs now exist to search the database in various ways. The search can be done on a textual level, for example, to locate and analyze hormone genes across several organisms. The search can also focus on various biological patterns like intron-exon junction, putative promoter sites, or splicing sites in nucleic acids, and drug interaction or secondary structure sites of proteins. The more sophisticated programs allow the biologist to communicate with the computers in a language close to biological English. Then let's see the application of computers in nucleic acid and protein sequence analysis. Many important analysis of nucleic acid and protein sequences are now routinely done by computers. For nucleic acid, their functions may include determination of mono, di, and trinucleotide frequencies, location of restriction enzyme cleavage site, as well as other sites that can be described as a pattern of nucleotides, location of various types of pair-rich region, then location of homologous symmetrices and diet both within and among molecule. Translation to amino acid sequences, determination of secondary structure for RNA molecules. Similarly, for the proteins, this function may include determination of amino acid frequencies, reverse translation to give nucleic acid sequences, location of possible restriction enzyme cleavage sites in the corresponding nucleic acid sequence, determination of secondary structure like alpha helices, beta sheets, and turns, location of hydrophobic and hydrophilic regions, and finally the comparison, both structural and functional, with other protein sequences. These are the functions which can be performed on protein and nucleic acid sequences. Then the other application may be the restriction map generation. Restriction map can be generated unambiguously from nucleotide sequence using computer programs. Map construction as one of their most often used functions. However, genetic engineers often construct a restriction endonuclease map from single, double, and sometimes partial and triple enzyme digest data prior to the availability of complete sequence data. This determination is normally combinatorial, and in all but the simplest of cases, there are millions of possible maps that must be considered to ensure that the best map has been chosen. A computer program called MAP has been developed by William Pearson, Johns Hopkins University, Baltimore, and IntelliGenetics. The map calculates digest lengths directly from band position on gels, takes into account constraining information like the existence of known sites and fragments, and presents a ranked list of all possible maps to the experiment. In this way, a genetic map, restriction map, can be easily prepared using these computer programs. Then let's see the other function, the simulation of molecular processes. Computers have been used to simulate many aspects of biological system. An example on a strictly structured level was aimed at solving the problem of maximizing the percentage of single insert of a linear fragment into an open plasmid that would then recircularize. A straightforward Monte Carlo simulation system was written based on a theoretical study to determine optimal concentrations of reactants and reaction times. Another program that simulates the structure to functional relationship of lambda phase system as it decides whether to reproduce in lytic or lysogenic matter. Researchers used the artificial intelligence based knowledge based system to build an completely transparent simulation system containing information about genes, proteins, protein product, and control loci in lambda. This system correctly predicts the behavior of lambda when various combinations of genes and control loci are mutated, including predictions of uncertainty and instability. The knowledge-based approach allows computer neurobiologists to examine and alter the biological information used by the simulation system within becoming computer experts. Experimental planning and debugging. The problem of planning and debugging both synthesis and analysis experiments is a difficult cognitive task. Experiment planning entails choosing a sequence of laboratory tasks to satisfy an experimental goal, whereas experiment debugging occurs when something does not work properly the first time. A general function system known as SPX uses skeletal plan method for experimental design. SPX uses a biologist described knowledge base to become expert in a particular area of molecular biology. Genetic researchers currently use it to design cloning experiments. An artificial intelligence system was added to help a scientist to analyze the faulty experiment for the following three types of problems. First, any technical error. The scientist added too much salt. For example, the scientist may add too much salt or did not run the centrifuge at the correct speed. Or technical selection here or technique selection here, it may include the scientist choose an exonuclease when he really wanted endonucleases, or he forgets that chaotropic solvents interact unfavorably with certain enzymes. Or the third way is the overall plan was faulty. For example, the individual component step of the experimental plan worked but did not solve the original problem. This in these types of problem, the system determine if design choices are valid, simulating if determine if design choices are valid, simulating the action of each step to predict changes in key laboratory parameters that may then be compared to actual results. In this way, technical errors are located and the exact nature and location of the flaw may be isolated. Then computers in medicine, computer technique in medicine. The application of computer technique in various fields of medicine is briefly described here. Let's see them one by one. The hospital information system. The medical informatics is a rapidly growing discipline. It seeks to organize and manage information in support of patient care, biomedical research, and education through the aid of computer and information network. A computerized hospital information system can establish consistent standard in the transmission and storage of data and continuously monitor all transactions. It provides easy access to valuable patient care information. The physician can have direct access to all the information of his or her patient through the use of computer. A hospital information system generally covers area like registration, administration or transfer, discharge, billing, medical record, index, wards, operation theatre scheduling, store or inventory pharmacy, diet, biomedical maintenance, payroll, accounts, etc. Then computers in data analysis in medicine. In medical research, large number of data is collected. This data is to be compiled, analyzed, and interpreted. For this purpose, certain statistical methods are to be applied. These includes calculation of standard deviation, standard error, application of test of statistical significance like the z-test, unpaired and paired t-test, and chi-square test. Statistical methods are time consuming. However, with the help of computers, large number of statistical calculation can be performed in a very short time. Several good quality statistical packages are available which allow the use of many more methods than is practical using traditional paper calculation. The examples are the Biomedical Computer Package or BMD, SPSS or Statistical Package for the Social Sciences, GenSet, EPInfo. These are the some example of statistical packages for biomedical research or analysis. Then let's see the computers in laboratory computing. The primary objective of a clinical laboratory is to provide accurate results in a short period of time. Laboratory analysis includes blood chemistry or hematology, we can say, photometry, microbiological studies, etc. Results must match with the patient identification details and should be valid. Quick access to laboratory system can contribute to efficient patient care system. Then let's see the computer assisted decision making, or we can say CMD. It is an interactive computer system that directly assists doctors with clinical decision making tasks. The system is intended to support doctors, complementing their natural abilities to make judgment with computers' vast memory, reliability, and processing capabilities. A general model computer assisted medical decision making has been developed. As here you can see that in the CMD, the first patient data is collected and it is put forward to the inference engine and compared with the knowledge base. Through comparison to the knowledge base, the system gives the probable diagnostic methods and suggests the treatment plans. Then let's see the use of computers in care of critically ill patients. Critically ill patients require large number of therapeutic intervention to optimize their chances of survival. For this, the variables must be collected frequently and the data derived therefrom made available to the clinicians and nursing staff. This results in a large quantity of information which may lose its significance unless the data recorded is presented in a clear manner. In this intensive care unit, it is now possible to computerize the total management of data recorded on the patient. The data management includes the entry, integration, and reporting of all vital signs, medications, intake and output volume, and laboratory values. Closed loop system for the direct computer control of the infusion of vasodilators has been developed. An intra-arterial cannula connected to suitable cardiovascular monitor provides the input signal to the computer. A pump which infuses the vasodilator drug to the patient is controlled by computer to maintain the arterial pressure within predetermined units. Then let's see the computer assisted therapy. The methods for planning, monitoring, and adjusting doses regimens of powerful and potentially toxic drugs like the digitalis and antibiotics like gentamicin have been developed. The physician can plan doses regimes by selecting a target peak total body concentration of a drug using computers. Then the application of computers in medical imaging. During the last decade, computers were commonly used for the high resolution image generation. Dedicated hardware and software is required to generate such images in CT scan, MRI, ultrasound, and gamma cameras. It is possible to integrate these workstations to the main hospital information system. Three-dimensional images of living human anatomy, regional physiology, and biochemistry in health and disease are in use. So these were the various applications of computers in biology and in the field of medicine. So let's conclude our discussion. Basically, the biology and computing both are changing rapidly. Although computers can store and retrieve large amount of data and perform complicated analysis in a very short period, the major limitation is that they lack decision making power and are not sensitive like human brain. Computing hardware will continue to get less expensive. Computers are becoming smaller and more powerful. Softwares will continue to get more expensive as the problem becomes harder. The introduction of artificial intelligence system will start to play increasingly an important role in managing the most precious component of high technology enterprise, the expertise of its scientists and engineers. However, at present, the data entry is a major hindrance for using computers by medical professionals. Whereas the computer viruses, these are basically a generic term used to describe any of a group of willfully destructive computer programs. Viruses can multiply with repeated use of a program and can be transmitted with the executable programs. They can destroy program files, data files, or erase all data from hard disk. There are antivirus programs available to remove a virus from a file. However, due to the newer introduction of viruses, it is difficult to completely eradicate these viruses. So these viruses, computer viruses, are basically harmful for the hardware and software of the computers and can destruct a complete system which is based on computers. So we need to take care of these computer viruses.